Hi, I'm Aiden Hernzer.
I'm Joaquin.
I'm Tuan.
And I'm Angel.
Our group's topic for the DNA project is restriction fragment length polymorphism, or RFLP.
RFLP is a tool used for DNA typing, which consists of four primary steps: extraction, quantitation, amplification, and capillary electrophoresis, or gel electrophoresis.
The technique was first developed by Alec Jeffreys in English Sciences in 1984 while researching hereditary disease. It wasn't until 1986 in the United Kingdom that it was first accepted in court when a man was accused of two sexual assault charges. He was later proven innocent of the crimes.
Extraction is the process of opening the cell's nucleus and isolating the DNA molecules in another solution. It is best to try and find as much DNA at the crime scene as possible, but contamination by other substances, such as hemoglobin, can provide a degree of difficulty in the later steps.
To undergo extraction, we must first get a sample of blood from a source. After getting our blood sample, the blood undergoes cell lysis, which breaks down the cell membrane, allowing access to the nucleus. This can be done by using detergent or enzymes, which will destroy the cell membrane, allowing for easier extraction of DNA. Next, the protein in the sample are removed as they can interfere with DNA analysis. This, again, is achieved by using chemicals or enzymes to remove the proteins from the sample. After that, the sample is then purified of other components. Purification can be reached by using methods such as phenochloroform extraction, which separates nucleic acids, or just DNA, from other molecules. Once that occurs, the DNA is assessed to determine whether the quantity and quality of the DNA is good enough. After that, the next step is quantitation.
Quantitation is used to determine if the sample containing DNA is human and not some bacteria or other organism. In order for the next step, amplification, to be carried out to its fullest potential, quantitation must determine the amount of DNA in the sample. This is due to the fact that amplification requires a narrow range of DNA input. There are many things that can be learned or used from quantitation. PCR, which Aiden will go more in depth later, will be able to occur, as well as DNA profiling and quality control.
Amplification is done by using a technique called PCR. PCR is the process in which of making billion PCR is the process of making billions of copies of the DNA sample, since the crime scene most likely does not have as much quality or quantity. It allows us to detect and identify gene sequences a lot easier by visual techniques. This process consists of heating and cooling the samples in a 28 cycle process.
After the amplification process is complete, many of the DNA molecules are all jumbled up in the mixture. The molecules are then put in a gel-like substance to be shocked. Since the DNA molecules, or nucleotides, are negatively charged, they shoot a current through the gel, and the smaller molecules will gradually make their way to the positive electrode, or the anode, which is placed on the opposite side, while the larger DNA molecules are not affected, but because of their larger mass, making them not move as far or not at all. After the DNA is collected from the capillary electrophoresis, or gel electrophoresis, it gets attached to a CE instrument and then put in a software in which the DNA profile can be made.
RFLP is analyzing the differences in people's DNA sequences by observing the restriction enzymes which are produced by bacteria to cleave parts of the DNA and are activated only by restriction sequences. After they are activated, they destroy bonds between nucleotides. These restriction sequences will be passed down among generations, and thus ancestry can be recorded based on these sequences. This is simplified and illustrated by the use of 2D bands on a sheet of paper that align in a column underneath a person's name. These bands are compared to those of suspects, victims, potential relatives, or other scenarios.
In order for the evidence to be individual through this technique, the DNA must be a 100% match of the suspect, victim, or etc.
The information obtained through these processes allows for convictions to be made with 100% certainty. It can also be used to track people's ancestry through their sequences and discover much more about their past. Although this technique is a phenomenal tool in forensics, its use goes far beyond that and shows the story of someone through their ancestors. As we continue to change and evolve as a species, we can look through these restriction sequences and remember how far we've come as a people.